Alrighty, welcome back everyone to part 2 of session 5 of Beyond Genes. This part focuses on heritable effects of diethylstilbestrol, or DES, the toxic pregnancy drug that was administered to many millions of pregnant women from the 1940s through the 1970s. And I'm pleased to introduce you to the chair of this session, Sue Robati. Sue is the executive director of DES Action USA and also the founder and president of the MedShadow Foundation. Now, Beyond Genes is a scientific conference, but when I spoke to Sue about the idea of focusing on DES, she had suggested a portion of the conference be more public facing, and that was a great idea. I'm so glad that we're doing that. And so we're shifting gears a little bit, and this is that is going to be this part of the conference, more geared towards a general audience. So with that, I am going to introduce Sue, and I'm going to turn myself off.
Okay, I'm up. I'm Suzanne Robati, and thank you for the intro, Jill. I'm so happy to be chairing it. We have an amazing group of panelists here today. I do want to say just one or two quick things because this is a mixed audience of both lay people and researchers, scientists, doctors. I just want to remind everybody that DES was prescribed to between 5 and 10 million pregnant women in the years between 1940 and 1971, when the FDA changed its indication and made it contraindicated for pregnancy. That doesn't mean that nobody's gotten it since then, but most of the exposure was over at that point. When DES damage was discovered in 1971 and published by Dr. Robert Herbst, it was hoped that the harm would be limited just to the offspring of the women given DES. That was not, unfortunately, what happened. Within a couple of decades, it became clear that the mothers who'd been given the DES were at a higher risk of breast cancer, and today we're talking about how the grandchildren are affected. One term I'd like to point out, and that is lay people most often refer to the women who were given DES as DES mothers or generation 1, and the DES daughters and sons as Gen 2, and the grandchildren as generation 3. Scientists and researchers refer to DES mothers as F0, the sons and daughters as F1, and grandchildren as F2, F referring to, I believe, filial. Anyway, just a point of clarification so that everybody speaking understands the same language. Anyway, our two main speakers will be drawing on the two major ongoing US databases that actually have DES data, live DES data. Dr. Linda Titus will be talking about the NCI Multi-Generational DES Follow-up Study, and Dr. Marianthi Kimojuli, I'm sure I killed that, will be reviewing her review of DES grandchildren based on the Nurses' Health Study too. After that, we have 4 shorter presentations from Carrie Christensen, Subra Mandal, Scott Curlin, and Michael Tournaire. We're going to take 1 or 2 quick questions after both Dr. Titus and Dr. K, I'm going to call her, hope she doesn't mind. And then for the next 4 shorter presentations, we're going to hold questions till the end, and then we should have plenty of time for questions. So I've asked Linda Titus to present first. Dr. Titus is presently at the Dartmouth Institute for Health Policy and Clinical Practice. In addition to her work in environmental and genetic risk factors for melanoma, breast and ovarian cancers, she has spent decades researching mechanisms and health outcomes related to diethylstilbestrol, to DES. She is a principal investigator of the continuation of DES follow-up study and has particular expertise in the development of DES grandchildren. So Linda, I turn it over to you.
Thank you, Sue. Here we go. I am going to talk about DES and focus on the NCI study. And obviously there are other studies that have been done looking at DES, and the Nurses' Health Study is one of them, and you'll be hearing about that soon. So let me get my slides up. Here we go. Sue made the title for me. Thank you, Sue. Can you hear me okay? Because I'm not using my headphones.
We hear you fine.
Good, thank you. So I'm actually at the Muskie School of Public Service now at the University of Southern Maine. I've retired from Dartmouth. I'm a professor emeritus there. This is the DES study group at NCI. Unfortunately, not everyone is still with us, but it's been a fantastic collaboration. So DES is a non-steroidal synthetic estrogen. It had many sources of exposure to the human population, but the one that we're concerned about really is when it was given to pregnant women. It has a very powerful estrogenic binding affinity. And the dose, I actually calculated, I think my calculations are correct based on Premarin, the range of Premarin doses given to women who are postmenopausal or experiencing menopausal symptoms. It's about 27 to 114 times 1 year's dose. So a pregnant woman was getting a huge dose, 12.5 grams cumulatively, if she followed the Smith & Smith regimen, which was popular at the time. It crosses the placenta and it concentrates in the fetal reproductive tract. So as Sue mentioned, the drug was given primarily between the 1940s and early 1970s. And it was thought that it prevented miscarriages or premature deliveries. It did neither. And in fact, later analyses of some of the studies showed that it made things worse. So at first it was given to women with a history of pregnancy complications. Later, it was just handed out with the thought that it would be helpful. And we don't know how many people were exposed, but possibly hundreds of thousands or even millions of offspring exposed in utero. It was given out in Europe, not just the United States. And interestingly enough, it continued to be prescribed to pregnant women, even though mammary tumors were seen in mice given DES during pregnancy. And I want you to kind of keep that in mind, because DES is one of those scenarios where the human situation and the situation for the mouse are very similar. And there were also two pregnancy clinical trials, one at the University of Chicago and one in London, and neither one of them showed that this drug was useful, but it continued to be prescribed. This is an ad from a major medical journal in 1957, basically saying, if you want a big, fat, healthy baby, you should take this drug. In 1971, the FDA banned the use of DES in pregnant women because it was discovered that the prenatally exposed daughters had an extremely high risk. This is a 40-fold increased risk of clear cell adenocarcinoma of the vagina or cervix. And this makes the association between smoking and lung cancer look almost non-existent. I'm exaggerating a little bit, but the fact is this is just a huge increase in risk that we don't usually see. It's a rare cancer in women of any age, and it was seen in very, very young women, post-adolescents for the most part, although I believe there was a 9-year-old. So in response to congressional pressure, which in turn was in response to advocates, DES advocates, the NCI sponsored a study in 1994. It recruited 5 study centers, all of whom had previously studied cohorts at their disposal. And these cohorts were combined, and then they were also expanded. So for instance, at Dartmouth, we had previous studies of women given this drug during pregnancy. So we found their sons and daughters by getting in touch with the moms, the first generation women. At other cohort and other centers, for example, DSAD, the daughters were being studied. And so we tried to find the moms by talking with the daughters. And similarly at Mayo, for example, there was a cohort of sons. So we found the moms from them. And we had quite a high number of participants in all these studies, over 2,000 exposed and unexposed women who were given DES in pregnancy, almost 6,000 prenatally exposed women, and about half that many, 2,600 unexposed women. And as far as the men go, it was a little bit smaller, where it's just a little bit over 1,000 exposed and 1,000 unexposed men. Still a relatively large study. So what we found in the NCI study of the first generation women, which as Sue explained is the group of women given DES in pregnancy, is that there's an increased risk of breast cancer. That's what was seen and predicted by the mouse model, which came before the human study. We also saw an increased risk of breast cancer mortality, but surprisingly, at least to me, no increased risk of endometrial or ovarian cancer. These sorts of studies had been done before. We were not the first, but we were probably the largest. A really important strength of this study that I should emphasize is that we had medically verified DES exposure or absence of exposure in every single person who participated in the study, and that's quite unique. We also confirmed the second generation outcomes, the outcomes that had been seen before in prenatally DES exposed women: infertility, higher risk of infertility, spontaneous abortion, preterm delivery, basically just adverse reproductive outcomes. We found some new things too: early menopause, grade 2 or higher cervical dysplasia, breast cancer at age 40 or more, and just recently we published a paper on pancreatic disorders and pancreatitis. Possible, just a suggestion that even pancreatic cancer may be a bit higher in the prenatally exposed women. We found, we did some psychosexual analyses too, and we found that the prenatally DES exposed women were less likely to have same-sex partners, weren't more likely to be left-handed, which we had expected, and were not at increased risk of depression or other mental illness overall, which will be interesting because you're going to hear a very different story from the Nurses' Health Study. But we did see a possible increased risk of depression or mental illness with low or very early exposure. In the men, we don't see as many adverse outcomes. No difference in overall cancer risk, maybe an increased risk of testicular cancer, which unfortunately will never be confirmed or refuted because the cohorts exposed to DES prenatally are now past the age of highest risk of testicular cancer. And we did see also in the men an increased risk of pancreatic disorders and pancreatitis. And that paper, again, was just published, I think, a few months ago. The men, however, do have genitourinary anomalies, and yet it seems that these abnormalities do not affect their ability to father children. We do not see an increased risk of depression in men, maybe an increased likelihood of same-sex partners in adulthood, and maybe an increase in left-handedness. And I'm saying maybe because the results were modest and not always statistically significant. So again, I want to emphasize that DES is one of those rare exposures where the human situation and the mouse outcomes seem to be very similar. So as I mentioned before, in the first generation, or as Sue pointed out, which would be FO in a laboratory, pregnancy is associated with increased risk of breast cancer. And prenatal exposure in women or females is associated with increased risk of infertility, vaginal adenosis, vaginal tumors, that's seen in the mice and in the women. And in the males, there is an increased incidence of cryptorchidism, epididymal cysts, and maybe of testicular cancers. So the question is, what's going on in the third generation? The animal models do predict outcomes associated with DES in that generation, the third generation, which would be the grandchildren of the women who were given this drug during pregnancy, or you could say the sons and daughters of the women who were exposed prenatally, or potentially the men who were exposed prenatally, but we did not study the offspring of men. So I can't address that question, and I wish I could. The mouse model shows an increased frequency of benign tumors of the ovary and adenocarcinoma of the uterus in this third generation, which would be the F2 generation in the laboratory. The mouse model did not show an increased risk of infertility, though, and we'll see whether we find that in the granddaughters. We don't really know what the mechanisms of intergenerational transmission might be in humans, but it appears that in animals, DES exposure alters gene expression. You've heard a lot about gene expression, some pretty technical talks, and it's possible that if prenatal DES exposure causes epigenetic changes, that they could be heritable, in which case we would see them in the grandchildren, and we would see them in the great-grandchildren too. So that's certainly a possibility. There are lots of mechanisms that are proposed and have been studied in the context of DES exposure. And the thing about gene expression is that it's not DNA mutation. You probably gathered that already from the previous talks. It affects the phenotype or the way or function, but not the genotype itself. It can result from environmental exposure. That's almost inherent in the definition. And it can be heritable if the exposure is early in the development of the germ cell line. And of course, when you have prenatal exposure, that's pretty early. It can't get any earlier than that. The mechanisms may include DNA methylation, histone modification, altered microRNA expression. I'm not a laboratory scientist, so I won't address any of those, but there's certainly been some great talks today from people who know all about that area. We're very expert. So these are some of the genes that are thought to be involved: HOX, homeobox, WNT, lactoferrin, and CFOS. But there is another explanation, maybe not in the mouse model, because the mouse model seems to be truly transgenerational, meaning that it goes out to what we would call the great-grandchildren, or the F3 generation. We don't study that. We haven't got that far. We have the first generation who were given the drug in pregnancy, the second generation exposed prenatally, and the third generation who are the offspring of those who were exposed or not prenatally. So there is another explanation that we need to consider, which is that DES is directly toxic to the primordial germ cells in the prenatally DES exposed individual. And that would mean that the grandchildren would be affected, but the great-grandchildren in subsequent generations would not. And we don't have the answer to that question, and I don't know when we will. It certainly won't be anytime soon. So the NCI, of course, wanted to address the questions that we could answer in the third generation, and we put together the NCI Third Generation Study. I was lucky enough to be the person who led that study on behalf of the NCI. It started in 2000, and we're just finishing up now. We're just starting to clean up the data now. And the eligible third generation participants had to be at least 18 years of age, and they had to be daughters of the prenatally exposed or the unexposed women who were already participating in our NCI second generation study. Now you notice that we aren't talking about the sons of the prenatally DES exposed or unexposed women, nor are we talking about sons or daughters of prenatally exposed men. So this is a definite limitation to our study. We don't know too much about the men. We didn't study them primarily for reasons of funding, and also because the effects of DES are so much more profound and widespread in prenatally exposed women than in prenatally exposed men. So our approach was, we went to the second generation women. We identified those who had daughters, which we were able to do, and because they had answered questionnaires to that effect, and we asked them for their daughters' contact information. Roughly half were willing to do that. And then we contacted the daughter with the information given to us by her mother and sent her a questionnaire. And some of these questionnaires were mailed, some were administered by phone, and some were done online. And we tried to confirm self-reported pathology conditions of interest through medical records or pathology reports, including things like breast biopsy or reproductive tract biopsies. We were aware that we had a very limited amount of data, not just in the size of the third generation cohort, but also because we weren't learning anything about the sons of the prenatally exposed and unexposed women. So we also asked the second generation women on their own questionnaires about their sons and their daughters. We didn't ask lots of questions, but we did ask them whether their son or daughter had birth defects on the 1997 questionnaire. And on several questionnaires, we asked about cancer diagnoses. I've grayed out some of those years because we haven't analyzed those data yet. That will be our next adventure. The third generation analyses then, just to be clear, come from two sources. We have the third generation daughters, the granddaughters, who are participating in their own study, self-reporting their own conditions. This includes birth defects and cancers, which we asked their mothers about, but it also includes hormonal factors, menstrual factors, use of oral contraceptives, reproductive outcomes, and things like that. And the second generation women, as I said, reported birth defects and cancer diagnosis affecting sons and daughters. So to confuse you further, this complicated study also has had 3 phases of data collection. We started in 2000-2001 with a baseline enrollment of those women who were at least 18 and whose mothers gave us their contact information. We then, in 2009, followed those women. We sent them follow-up questionnaires to update their outcomes. And we also enrolled a new group of women who had turned 18 between 2001 and 2009. And then just recently, 2019 and 2020, we followed the women who had participated in 2001 and 2009 and identified yet another group for the baseline participation. And as you might imagine, though, that group is smaller now because most of the women were born before 2000, 2001, most of the third generation women. So for the first group, we've got 898 daughters, and we had pretty good participation. Second group, we also had pretty good participation in the new baseline study. We didn't do as well as we wanted to in the follow-up study of the women who had participated in the baseline study in 2001. So we're going to look at birth defects, hormone reproductive outcomes, and cancer diagnosis based on the 2001 and the 2009 questionnaire combined. And then separately, we looked at the follow-up women because follow-up women, women who participate in follow-up may be a little different from baseline. Baseline, we always worry that women are enrolling in the study because they have a condition that could be associated with the exposure. So we want to look at follow-up to see whether that association persists. So we have the mother's reports of birth defects and the mother's reports of cancer diagnoses from several questionnaires. And this is what we found in terms of birth defects when we asked the mothers, the second generation women, prenatally exposed compared to prenatally not exposed, about birth defects in their sons. And you can see that we have a slight increased risk, or it's a modest increase of 1.5, so about a 50% increase in risk of any defect. None of the specific sites of anomalies were statistically significant, but I think that's because we have very, very small numbers, so we're underpowered to look at these individual outcomes. But there certainly was a suggestion of an increased risk of genitourinary or penile testicular anomalies. We asked the mothers also, prenatally exposed and unexposed, about their daughters' defects, birth defects. And here we have a stronger association, an odds ratio of 2.4 more or less, so it's more than a doubling of risk. And here we see also a significant association specifically with the group of anomalies associated with the heart and with the skeleton. Unfortunately, we can't draw any conclusions about either of those because when we looked at the general population, heart defects in newborns and children, we realized that what might be going on was and underreporting by the unexposed mothers. And similarly with the skeletal defects, many of them were hip dysplasias or hip anomalies. And I think that because of the reproductive tract anomalies in the mothers themselves, some of these, if not most of them, may be due to birth trauma. So we're not sure we're looking at actual effects of DES. I'm not going to rule it out, but I can't say with certainty that that's what's going on. Now, interestingly, when we ask the women, the third generation women who are participating in their own separate study, they didn't report that much in terms of birth defects. None of these associations were statistically significant, not even the one in association with any defect. So in the men, what do we conclude about third generation men? Well, we really don't know whether we see an increased risk of birth defects at specific sites. That could be a problem of statistical power. It does seem that there's something going on there. And in the women, we do see an increase overall in birth defects, but it's due mostly to heart defects and to the hip anomalies, the skeletal anomalies. And we're not sure whether those are direct or indirect DES effects. When I'm referring to the skeletal anomalies when I say that, because the heart defects we think might have been underreporting by unexposed mothers. So now we're going to look at what the third generation women reported themselves in terms of menstrual characteristics, fertility, pregnancy outcomes. Obviously, when we're thinking about DES and the second generation effects of prenatal exposure, these are the areas in which we have a great deal of interest. We did not, however, surprisingly see any difference at all in average age at the beginning of menstruation, 12.5 versus 12.7 in unexposed. It's pretty close. And the mean age of regularization of menstrual cycles was showing a little something. Here we have women who are DES exposed or whose mothers were DES exposed, reaching regular menstrual cycles, which they could predict within 5 days at a slightly older age than the unexposed, and it was statistically significant. The report of irregular menstrual periods, not surprisingly, given what I just showed you, suggested that the daughters of prenatally DES exposed women were at slightly increased risk, 1.2, that's about a 20% increased risk of having irregular periods and of suffering for, or going a year, maybe it's not suffering, but going a year without a menstrual period. Very similar increases in those odds ratios. Infertility, there was a suggestion there that infertility was higher in the third generation women who are DES exposed. As you can see, it's 1.5, was not statistically significant, but it was suggestive. And in the 2001 data, which we analyzed separately and reported previously, there were more worrisome things going on in terms of infertility. And we thought we saw an increased risk, especially in women who were age 35 or more. It did not really show itself when we combined the 2001-2009 baseline data several years ago for analysis and reported them separately. Medical help for infertility was also slightly higher in women whose mothers were prenatally DES exposed. But again, it was not statistically significant. Infertility, did I just show that? This is the follow-up, sorry. So these are the follow-up data. Remember when we did the 2009-2010 data collection phase, in addition to enrolling new baseline women for the baseline questionnaire, we also sent a follow-up questionnaire to the women who had participated in 2001. We did not see an increased risk of infertility in the follow-up women. And this is exactly the kind of phenomenon that can happen that I was discussing before, where sometimes people enroll in a study because they do have a condition. And here we did not see it at follow-up, which was a bit disconcerting. We're glad we didn't see it, but it does call into question what we saw at baseline. There was, however, an increased likelihood of seeking medical help for infertility. It wasn't statistically significant again, but we have small numbers here, so that's not terribly surprising. So in summary, for just the menstrual characteristics and infertility, the daughters of prenatally exposed women were more likely to have irregular menstrual periods and to reach regularity in their menstrual periods at a slightly later age. It's not clear what's going on with infertility. That's something we want to take a close look at moving forward. Reproductive outcomes looked very similar for the exposed and unexposed. This was also true for specific adverse outcomes like miscarriage or ectopic pregnancy, but we might see an increased risk of preterm birth and ectopic pregnancy. It's the kind of thing we need to pay close attention to as we move forward. In 2001, the majority of women had not been married yet or started their families. In 2009, most of them had, but I think with this latest follow-up, we should get a little closer to answering this question because most of the women in the cohort will have probably completed their families. So now we're looking at cancer, which of course is also an area of big interest because of the very, very high risk of clear cell adenocarcinoma of the vagina or cervix in the prenatally DES exposed women. So we looked at the sons and the daughters based on the mother's reports. Now that SIR, I don't know if you know what that is, but in this analysis, because the numbers were so tiny, we compared them, instead of doing an internal comparison, we compared them to the number that would be expected in the general population for that gender, that sex, in that age range. And as you can see, we didn't see anything that was worrisome in terms of increased overall cancer risk. There's a suggestion of 1.3 in the DES exposed daughters, but it's 1.6 in the unexposed. And so we're not, there's nothing there that could be remotely considered certain. And then we looked at the third generation women's self-reported cancers. This is based on the 2001 baseline data. We did not combine the 2001-2009 baseline data yet for analyses because what we saw in 2009 was different from what we saw in 2001. I knew that the 2020 and 2019 data would be incoming, and I thought it would be best to wait. I did not want to report a seesaw. Yes, we see it in 2001. No, we don't in 2009. Yes, we do in 2019. That's just not good science. So we decided to wait until we had what might very well be our final round of data collection. So here we saw some indication that cancer risk overall might be exposed in the elevated in the daughters of the prenatally exposed women. It's about double, but again, it's not approaching statistical significance. However, we saw something very alarming with regard to ovarian cancer. There were no cases in the unexposed. In the DES exposed granddaughters, these are, again, the daughters of women who were prenatally exposed to DES. We saw a nearly 15-fold increase in risk when we compare those women directly to the unexposed, to the general population. And it's only 2 cases. So we can't draw firm conclusions, but it's very worrisome. I want to show you at the bottom of the slide what happened with leukemia, though. We saw when we compared the exposed and the unexposed to the general population, we saw an increased risk of leukemia in the unexposed women, the daughters of unexposed, DES unexposed women, when compared to the general population. This is the kind of statistical fluke that we sometimes run into. And we can't be sure that that's not what was going on with the ovarian cancer. That could also be just a statistical anomaly. However, if you remember, the mouse model suggested ovarian tumors. in the third generation or the F2 generation. So this is something we're definitely concerned with. As for those ovarian cancer histologies, the ones that were self-reported, one was a serous borderline metastatic tumor at age 20. One was a mucinous borderline at age 22. These are very young cases. Ovarian cancer can affect very young women. We had very young women in this study. We couldn't have seen it in older women for that reason, but it's still concerning. And both of these were confirmed by the medical record. Now, we also asked the mothers, of course, about their daughters' cancers, and the mothers also reported 2 cancers in their daughters, one of which had already been reported by a daughter in the third generation study, the other, which obviously wasn't because it occurred in a very young woman, and that was a juvenile granulosa cell at age 7. I have to say that when we're talking about prenatal exposures, we worry about very young ages of cancer diagnosis, and that's one of the things that's worrisome with these data, is that it could indeed be a DES effect. We may never know. We may never have the answer to this question, but it's certainly worrisome. So we compared the self-reported ovarian cancers to the general population, and I wanted to just show you that there were only 0.14 expected ovarian cancers in the general population for this age group. And then we thought, well, you know, not everybody participated in the third generation study. We had really good participation once we approached the third generation women, but if you remember, only about 50% of the mothers gave us permission to approach their daughters and contact information so that we could actually reach their daughters. So we thought, let's recalculate that standardized incidence ratio, comparing the number of ovarian cancers that we saw to the population, the general population, assuming that everyone who could have participated in this study did. And although it wasn't 14.68, it was 6.61 and statistically significant. So we still have a highly, it's very significant and disturbingly high association here between a third generation DES exposure and an outcome of ovarian cancer. So the leukemia, just to go over, we did the same thing, and we pretended that everybody had participated in the study, and now it's a 5-fold increase, and only 1 case was expected. Don't know whether this is a fluke. I mean, I assume it is. But as a summary of the cancer outcomes, we have very little evidence of total cancer, overall cancer risk in the sons or daughters of women who were prenatally exposed to DES compared to the sons and daughters who were born to women who were not prenatally exposed to DES. We do, however, have an apparent excess of ovarian cancer affecting very young women who are daughters of prenatally DES exposed women. And we have this weird anomaly in our data, which is an excess of leukemia in the unexposed daughters when they're compared to the general population. So as an overall summary, sort of looking back at everything, there appears to be an overall increase of birth defects in men based on the mother's reports, but it's inconclusive for specific types of birth defects. There's an overall increase of birth defects in women based on the mother's reports, and for heart and skeletal defects. Again, I'm not sure whether that's a direct or indirect effect of DES, and we did not see that when we asked the daughters themselves, the third generation daughters themselves, but of course that's a subset of all the daughters born to the second generation. We did see increased likelihood of menstrual irregularities and later regularization of menstrual periods in the women born to those who had been prenatally exposed compared to those who were born to women who were not prenatally exposed. Pregnancy and live birth seem to be similar, possibly an increased risk of infertility and ectopic pregnancy and preterm pregnancy, but we need to sort that out with the next round of data collection, and I hope we will. So we did not see overall increased cancer risk in women or in men, but we did see a possible increased risk of ovarian cancer, although the numbers are very small, with a total of 3 cancers when we combined the mother's reports and the daughter's reports. And what's disturbing about that, of course, is that it aligns with the mouse model. So I say le dor va dor, which is Hebrew, or from one generation to the next. Lessons for the future. This DES is a horrific example of medical mismanagement. Even the results of clinical trials were disregarded. It is a model of intergenerational endocrine disruption. Don't know whether that will continue down the generations. Don't know whether it's heritable, and we need to follow the third generation forward to try to figure out whether these reproductive outcomes and this unfortunate ovarian cancer outcome will continue to the next generation. This is the group of study investigators. People have moved around a bit, but, and unfortunately, Dr. Kaufman is no longer in this world. Dr. Adam, who's in his mid-90s, is still collaborating with us. These are my colleagues at UMass, Boston University, University of Chicago. Dr. Hoover at NCI has since retired. Sorry to say, we miss him. And Rebecca Troisi is still leading the study from NCI. Thank you.
Thank you. Thank you, Dr. Troisi. That was
Titus.
Titus.
I'm sorry, you said Troisi. I'm sorry, Titus, of course.
It's okay. We have been accused of being sisters.
I'm going to ask you one quick question, and then we'll go on to the next speaker. My question is, you used a phrase final round of data collection, as in the study would be. It implied the study has an end date. Does it have such an end date?
Yes, the end date was September 20th of this year.
And is that the final round forever?
That's the final round of data collection forever, so far as I know, yes. Yeah.
Well, that's sad. Okay, and I think, Jill, did you have a question? Yeah.
Yeah, just a quick question. There are a number of very good questions in queue. If you could also respond to those online in the Q&A box. I just have a quick question about the self-report of the third generation. Yeah. So did that necessarily exclude the granddaughters who couldn't self-report owing to intellectual disability or death?
But would naturally do that, yes. Absolutely.
Well, then could that help explain maybe the leukemia? I mean, if someone had died and they can't self-report, they're not going to show up in the data. I mean, similarly, if they're like my daughter, right, who is nonverbal and autistic and intellectually disabled, they can't report, and they might have, additional, issues that, would have shown up if it had been like an exam or if the parent had reported.
Right. And I get your point, but there's no reason to think that the deaths from leukemia would have been greater in the exposed group than in the unexposed group. And we also have the mother's reports, and we did not see an excess of leukemia in the reported by the unexposed mothers for their sons or daughters. So I don't think that's an explanation in this setting, Jill. I mean, obviously, when we're studying some cancers as outcomes, death is in addition to being a tragedy, it also inhibits our ability to understand the connection between an exposure and the disease.